I apologize, we're going to have a full day today because of my traveling last week, but I think it's pretty interesting and related material. Kai waited to ask a question until after class last week, which I think he should ask to finish the discussion about transplantation, and then we'll move on to the rest of the week. Go, Kai. What did you ask me after class last week?
About whether we can relate religion to the topic.
Right.
Because the main concern that I had was because I go to church and then from what point what considered as life. So it's embryo and things like that can be considered as life. So and different countries have different laws. For example, 18 weeks and certain weeks, different countries different idea. For example, China and Korea, we have different definition of what can be considered as life. And when it comes down here, some people say it's brain, some people say it's spinal cord. So aren't we basically killing someone to save someone? So that's moral and religious issue that can be considered.
So that's important. That's especially around embryos. What can anybody think of anything else that would be an ethical concern here, specifically around religion? Go ahead, Nancy.
Can I just use my religion as an example?
It's better to talk about your own than somebody else's.
Yeah, so as a Buddhist, we really emphasize going into the afterlife and stuff like that. And not having a body, we like to preserve body parts, and so not having a part of your body can, it'd be it's like going up to heaven without a liver or something like that is kind of a big deal. And so my mom especially is very against donating organs or donating blood. And I guess also cultural backgrounds, my parents see donating blood or something as a, I don't know, like in Vietnam they sell blood for money and stuff like that, so they're really against that because it's kind of lowering yourself in some way, and they're afraid that of me dying.
If you go down Main Street in Lewiston, on the opposite from Sam's, there's a plasma donation center where you can get paid
to donate blood.
Yeah, so that happens
So
in this country as well.
yeah, they're just so against it.
Okay, that's good. I didn't think about Buddhism. I was actually thinking about Jehovah's Witness.
They can't accept blood transfusion.
Right.
I just had a question. I know that they can't accept blood blood transfusions, but do they have something against organ transplantations also?
I won't speak. I assume so, but because blood would go with it, but I don't actually know specifically the answer to that question. Rachel, did you have a question?
In my biology class last semester, or 2 semesters ago, we talked about also Islam and not accepting pig or pork parts.
Oh, right,
Which is
artificial
not-
heart valves. and
Yeah,
stuff like that.
which is not, it's related, it's still transplantation.
Angelique.
But with Islam, it's not just artificial heart valves, it's the ones that are made out of pig. And that
Yeah.
goes back to pork, so it's not just all artificial heart valves, just to clarify that.
Yeah, so I'm Muslim, so I could probably clarify a little bit. So yeah, it's mostly we're not really supposed to eat pig, but that kind of stems from, so pigs are actually pretty clean animals on the inside when you look at it, but just the fact from not eating pig kind of stems from symbolically they were always kind of seen as a dirty animal because they were always in the mud and everything like that. So I guess it's kind of more open to interpretation of how you're looking at it. But yeah, a lot of conservative Muslims would definitely reject
So
pig
what
parts.
you're saying is there are lots of different kinds of Christians, there are lots of different kinds
Yeah.
of Muslims, there are lots of different kinds
Yeah,
of
it's not just one
Buddhists and
variant.
Jews,
Yeah, so in Judaism, they want to bury the body as quickly as possible. But they say if you can, take an organ to save a life, and that delays the body, the burial, that's okay.
Okay, good. So we covered many of the world's religions and their contributions to organ transplantation. I wouldn't say all, but I wanted to make sure we got that in. Okay, so the reason that we have so many lectures this week is because I went to a wedding last week. This is not on Lyceum. This is me with the groom who was wearing a kilt.
Did they give you?
What are you wearing?
That is a bunch of stuff I found at a used clothing store in Boston. Actually opened the same picture. There's the groom. There's the bride.
Wow.
My cousin. So
What was the theme of the wedding?
steampunk,
Oh.
which I'd never heard of before, but it was fun. Anyway, okay, so mucosal immunity. These materials come from a close colleague of mine at Pfizer by the name of Kara Williams. She heads our in vitro biology group. She was trained in asthma and mast cell biology, and if I can answer any of your questions, I will direct them to her. So we'll start with mucosal immunity, touch on memory briefly, which has come up in other lectures, and that'll set the stage for the vaccination discussion. And then depending on how that goes, we'll get a start on the vaccinations lecture. And I think at the latest, next week we'll be caught up. The second exam I posted last night on Lyceum, and again, if you can send it to me by next Friday, that would be great. Okay, so the mucosal immune system, obviously, this is actually one of the things we talked about in the very first lecture about innate immunity and where we interact with the environment. Obviously, the malt covers all our mucosal, or basically the wet epithelial surfaces of our body, so GI tract, nasopharynx, genital urinary tract. And we'll touch on most of those today, and then the salt, the skin-associated lymphoid tissues. We'll talk about how there are inductive, or basically locations of naive immune responses, and then effector sites where mature immune responses happen. Inside the galt, we'll talk about all the different structures and special cell types are involved with that, and then spend a few minutes talking about soluble IgA as the most important antibody in the mucosa. And then what we learn in the galt can be applied to most other associated lymphoid tissue within the malt. So let's give some specifics there, and finally differences with the salt. Okay, Kara gets better pictures than I do, so guys, we got lots of color today. Okay, so the malt is mucosal associated lymphoid tissue, galt is gut, malt is bronchi, malt is nasopharynx, and salt is skin. An inductive site is where the antigen is first encountered and where the primary adaptive immune response is generated. In the galt, these locations are Peyer's patches, the appendix, and diffuse collections of lymphocytes and antigen-presenting cells just under the gut epithelium. And there are inductive sites in the other tissues as well. Then the effector sites are a specific area where the effector lymphocytes are dispatched after the immune response. So, let me open the door. Anybody hot? So the inductive sites are specialized structures that are akin to lymph nodes, and then after immune response has occurred, the T and B cells redistribute throughout the tissue so that they are most likely to come in contact with antigen. And one important, interesting example of a mucosal effector site is the salivary and lacrimal glands. Why would that be a good place for mature T and B cells to go? So what do salivary glands and lacrimal glands make? Right? So what could you put into those saliva and tears that would interact with pathogens, if not cells? soluble immune effector molecule made by B cells. Huh?
Antigens? You get it?
Antibodies, yes.
Antibodies,
There you go.
not antigens.
So,
okay.
so you make a lot of soluble antibody, especially IgA and IgM, that are secreted into the mucus lining your gut, as well as tears and saliva. Okay, so as I just said, the mucosal inductive site is a specific immune structure. It's covered by specialized cells called M cells, which are good at transporting antigen. We'll talk more about those in a minute. And then you have these immature immune follicles, just like in a lymph node, which contain antigen-presenting cells, naive B and T cells. Just like lymph nodes, these naive lymphoid follicles are supplied by the high endothelial venules. So T cells and B cells are randomly recruited out of the circulation into these lymphoid follicles, just like in lymph nodes and other lymphatic tissue. So in the malt, like Peyer's patches, these M cells actually have a very specific invaginated structure, such that the immune cells actually cluster in under the surface of the mucosa, so that they are in very intimate contact with antigens getting transported out of the gut. So after an immune response occurs in the inductive sites, those T and B cells can then migrate out to a lymph node and undergo a germinal center reaction, producing mature and effector B and T cells, which then migrate back into an effector site where they can reside and secrete antibodies into the mucus or saliva or tears, and have a chance to interact with more of the same antigen through presentation by high concentrations of antigen-presenting cells. So very different structure. Any questions? But these sites are distributed throughout different sections of the gut. So, just a wave. Okay. So the gut has two jobs: absorb nutrients and repel pathogens. So, and we have to. We've actually discussed it several times. It's kind of amazing that we can eat everything that we can eat, which we know we have TCR and B cell receptors specific for all that foreign stuff, and we can be full of 2 pounds of pathogens, and yet we don't, most of us don't develop inflammatory bowel disease. So there's an incredibly complex balance between nutrient absorption and immunosuppression and effective immune responses. So it's not that surprising that things like celiac disease, Crohn's disease, ulcerative colitis occur in that environment. So this is another depiction of those inductive sites. Those of you who have taken physiology know that the intestine is lined with villi, which increase the surface area of the gut and increase the ability to absorb nutrients. Under the epithelial layer in the villus, there's a lot of loosely distributed lymphoid tissue, which includes T cells, B cells, and antigen-presenting cells. But this isn't the site of the lymphoid follicles, basically, those inductive sites, which have a specific structure because they have the M cells to transport antigen, they have dendritic cells, and a germinal center. So there's lots of lymphoid tissue in the gut, but the location where those primary germinal center reactions happen is in the follicles under the M cells. So when there is a pathogenic or inflammatory response to something in the gut, then that germinal center, that isolated lymphoid follicle, can mature, just like in the lymph node, then you get class switch recombination, somatic hypermutation, all the things that happen in a lymph node normally undergoing a response to pathogen. So you get expansion of T cells and B cells, production of those effector T and B cells, which can then, after effectively responding, recirculate to other locations within the gut. Ian?
So an ILF is just kind of a modified villus that has M cells.
Basically, yeah. Okay. So just like we talked about in the early lectures, the health of the gut epithelium is key to maintaining the barrier to all that foreign material. So in order to create an infection, an invading pathogen has to compete for nutrients and living space with all the normal, healthy, commensal probiotic organisms we have. They have to avoid getting swept away by mucus that the goblet cells lining the gut make. They have to evade all those secreted antimicrobial components of the innate immune system that we talked about, like lysozyme and other molecules. They have to evade any specific antibodies that we've previously made that are secreted into the lumen. And they have to actually counteract a negative charge that is associated with those cells. They have to get past digestive enzymes. They have to avoid antimicrobial proteins produced by Paneth cells, which we'll talk about in a minute. And they have to avoid activating TLRs on the epithelial cells. So this is a sort of exhaustive list of all the immune components that help most of us maintain gut equilibrium. Okay, so the lamina propria, that region of tissue under the epithelium, is the tissue-facing surface of that epithelium. And it's home, as I said before, to a great density of immune cells. In fact, the gut as a whole is the biggest lymphoid organ of the body. And so these cells are not only responsible for maintaining an appropriate immune response to pathogenic organisms, but also maintaining suppression of an inflammatory response that would compromise the gut epithelium. Because you can think about the fact that if we had an inflammatory response to every new pathogen that we ate, that we would get cell death, and we would affect the health of the gut epithelium and then allow infection to take place. There's lots and lots of conventional alpha beta T cells, CD4s and CD8s, as we've talked about before. Also quite a few gamma delta T cells and some other unconventional T cells we'll talk about later. Okay, so the antigen sampling happens in those intestinal follicles that we could talk about. So they're defined by the presence of those M cells. So they can occur in large clusters, which have the name structures, the Peyer's patch and the appendix, or they can be in isolated small groups. There is specialized epithelium associated with those follicles, which includes flat sections of gut epithelium and M cells. So those are the structures that are specifically designed to facilitate transport. So this is the conventional view of the absorptive portion of the gut with the villi, the circulation underneath, and it's not clearly depicted here, but this space, the lamina propria, is full of immune cells here. The base between the villi is the crypt, and as well as specialized innate immune cells called Paneth cells that we'll talk about in a minute. And then in isolated patches, there are those isolated lymphoid follicles. So I mentioned a couple of times the M cells. So these, I think their origin developmentally is epithelium, but they function very much like phagocytes. They're specialized for transporting particles of various sizes. They have, they don't have the microvilli and glycocalyx that the normal cells lining the gut do, but they use all the tools of the macrophage to bring substances out of the gut, which includes macropinocytosis, phagocytosis, and endocytosis. There are certain viruses and bacteria which take advantage of these cells as a passage into the body. So I talked about all the innate immune mechanisms that keep bacteria and viruses at bay and keep them in the gut. These bugs have adapted. They say, hey, M cells are really good at bringing substances in through the epithelium into the body, so they use receptors on the M cells to get through the gut barrier. Obviously, then they have to have other mechanisms for avoiding the immune system. They aren't antigen-presenting cells, though. They don't present antigen like DCs and macrophages. Okay. Let's keep it G-rated here. Okay, so this is what they look like on electron microscopy. So you can see the surrounding epithelium in this flat section of the, so these are villi. This is that flat section of FAE cells and M cells. If you blow that up, then you can see the individual cells of the flat epithelium and the M cells, and then blow that up further. That's the surface of an individual M cell where the antigen transport is going on. Any questions about so far?
Do E. coli, sorry, are E. coli like infections particularly bad because we recognize E. coli usually as like okay in our gut, but if it has that mechanism to get in, it is rather damaging, or?
So E. coli are interesting because we have tons of E. coli all the time. There's lots of E. coli in the soil. There's lots in the guts at all times. There are certain strains that cause disease when food is infected. It's really, different strains of E. coli have different abilities to adhere and invade the body. They don't have those specialized mechanisms of using the M cell. But it's basically particularly pathogenic or particularly high concentrations of E. coli that cause food infection. And some outbreaks of foodborne disease result from an actual infection. So you eat meat that has a lot of E. coli of a toxic strain, and you end up being infected with that of E. coli and becoming septic. Whereas when you eat a meal and you say, I got food poisoning because you vomit right away, that's actually an intoxication. You're not getting infected, you just have gotten so much toxin, so much LPS and TLR that you have an acute response to all the toxins associated. So there's, E. coli can cause both, because they have a lot of LPS that can cause a lot of innate immune response, but there are also strains that can cause a chronic infection and then become septic after invading the epithelium. Which is, why it's good to have healthy, normal flora to prevent opportunistic pathogens from getting involved. So, a few slides ago, I mentioned Paneth cells. These live down towards the bottom of the crypts between the villi. Again, just like the M cells are derived from epithelium but function like macrophages, the Paneth cells are derived from epithelium and they behave like neutrophils. They respond to PAMPs and DAMPs in terms of producing specific responses, specific innate immune responses. They secrete a number of antimicrobial molecules, which we'll talk about in a minute. And they live basically in the base of the crypts where there are stem cells that constantly replenish the epithelium. And the thought is, I don't think there's any great proof, but the thought is basically this could be a place for, down in the bottom of the crypt, where the digestive contents is not moving continuously, this could be a place where an infection could lodge.
So is the crypt just the area, the place where the villi split?
Yes. Yeah. So these are not necessarily associated with the lymphoid follicle like the M cells. The Paneth cells are distributed in the crypts, just as another component of the innate immune system. Okay, so way back in the beginning when we were talking about innate immune responses, neutrophils make a lot of defensins, and so do Paneth cells. So these are hydrophobic peptides that are good at lodging in the membranes of cells, which leads to their lysis and killing. They also make some digestive enzymes like lysozyme and phospholipase, which are good at breaking down lipid-based membranes, lipids and proteins in membranes. So this is just another, here's the cartoon. Basically, the epithelial stem cells are close to the bottom of the crypt, and we know that gut cells divide very rapidly. They're constantly replenishing the surface of the gut. This is why chemotherapy makes people sick to their stomach, because it kills the rapidly dividing epithelial cells of the gut. So those Paneth cells are sitting down in the bottom of the crypt. They're full of granules and all those digestive enzymes and defensins. Okay. So we've talked about the M cells, the Paneth cells, the general structure of the lymphoid follicles. We'll talk briefly about dendritic cells. Dendritic cells are not especially, they don't have special names or origins in the gut. These immature dendritic cells are functioning quite similar to dendritic cells in any other tissue. They migrate through the use of either CCR6 or CXCR1. So these are two specific chemokine receptors. So they're gradients of CX3CL1 or CCL20 that move these dendritic cells through the tissue. They're particularly concentrated in those lymphoid follicles, but also are distributed throughout the normal villi. They're very good at picking up antigens that M cells move across the gut epithelium, and they can also actually extend their dendrites between the epithelial cells of the gut to sample antigens that are passing by. So there are these CX3CR1 that are distributed throughout the gut, and then the ones that express CCR6 are mostly found in the domes of the higher patches, but they basically perform the same functions. So in general, dendritic cells live in the tissue in an immature form, so they don't have very big dendrites. They don't have a lot of class 2. They don't have a lot of antigen presentation capacity. When they take up antigen that stimulates their pattern recognition molecules like TLRs, then they become a mature dendritic cell, expressing lots of co-activators for T cells, as well as changing their chemokine receptor expression so they can move into lymphoid follicles, and then become an effective antigen-presenting cell that can either present antigen to T cells or produce cytokines to stimulate B cells and NK cells. So this happens everywhere. This is not specific to the gut. So the important function, as you can imagine, as we've been talking about today, the balance of regulation in the gut is particularly important. So we have all those dendritic cells that are receiving antigens from the M cells. They have their own dendrites in the gut sampling antigens. Most of that stuff going by is foreign. So they're presenting a lot of foreign antigens all the time to T cells. And in the absence of inflammation, those immature dendritic cells basically are presenting a lot of foreign antigens to T cells and causing anergy. So basically, they're giving signals to the T cells that tell them not to proliferate. And it's not entirely clear what's special in the gut that anywhere else, if you add that same antigen into your skin or put it somewhere else in your body, you would have a pretty robust inflammatory response. But in the gut, the cytokine and cellular environment is such that most of those foreign antigens are tolerizing T cells. They're saying, yes, I see it's your antigen, but don't respond. Yes, I see it's your antigen, but don't respond. And historically, one mechanism that allergists and immunologists use to tolerize allergic people is actually giving them small doses of their allergen orally, because it's actually possible if you're allergic to eggs, or if you're allergic to cat hair, or if you're allergic to dust mites, whatever, that feeding small amounts of that antigen, actually you can tolerize the specific T cells because of the sorts of antigen presentation that happens in the gut. Nick, go ahead.
When there is inflammation, then like, since inflammation is like basically signal of body, is it better to remove like if there's like inflamed in small part, is it better to remove it and decrease the pressure inside the skin, or like inside the inflammation, or is it better to just like leave it as inflamed?
You're asking whether you should pinch a pimple.
No.
Well, that's what it sounds like. So you're saying,
If there's inflammation.
yeah, so usually, so Jenny is a good example. She gets these cysts on her feet that between her toes, and so there's a site, it's actually sterile inflammation, and it doesn't resolve unless I drain it. So in general, pockets of inflammation or infection of a significant size need to be drained. That's a broad oversimplification. So yes, pinch a nipple.
Okay.
Anyone else? Okay. Right, so they tolerize the T cells. If there's enough inflammatory signal in the form of inflammatory cytokines like TNF, then those antigen-presenting cells, the DCs, can mature and now present antigen. And as I said, it's not totally understood, but the vast majority of this antigen presentation of gut contents is tolerogenic, not inflammatory. And as we've talked about in our T cell class, the type of cytokines made by the dendritic cells determine what kinds of T cells you get. So naive T cells can differentiate into Th1 cells. If they see interferon gamma, they can differentiate into Th2 cells if they see IL-4, IL-13. And the gut immature dendritic cells are particularly good at making IL-10. And that results in basically regulatory or suppressor T cells, these energized T cells. So this is just a demonstration that there are lots of different kinds of mature dendritic cells, and depending on what cytokines they respond to determines what kind of cytokines they make, and therefore what type of T cell response. So if you have a parasite in your gut, the adaptive response would be to make IL-4 and IL-13, so you make IgE to clear that infection. If you have a virus in your gut, you're going to have a Th1 response. If you, and so on and so forth, depending on what sort of antigen in an ideal world causes the inflammation in your gut. So this is just a computer colored electron micrograph showing some of the dendritic cell dendrites extending out and holding on to some foreign particle in the gut epithelium. Okay, so we've talked about the antigen-presenting cells, the cells of the innate immune system, general movements of lymphocytes. So we'll spend a little bit more time talking specifically about the lymphocytes in the lamina propria. So again, they're distributed loosely under the epithelium, and then they're also organized into the follicles. So there's a very high number of B cells in the lamina propria that have differentiated into plasma cells, and the vast majority of those are making IgA. So there's a huge population of B cells making IgA to things we've seen previously in the gut. So we're constantly secreting large quantities of IgA into the gut to help control pathogens there. I'm going to skip this actually. It mentions some specialized T cells that are observed in the gut. Nobody knows for sure their exact function. If you're interested in reading about intraepithelial lymphocytes, you can, but I'd say they're not. Just knowing that there are some unique lymphocyte classes in the gut serve that purposes. Okay. So, as mentioned, 70 to 90% of the B cells in the lamina propria make IgA. The remainder predominantly make IgM, with a few IgE and a few IgG producing cells. Obviously, if you're a person with a lot of gut allergy or are chronically infected with parasites, you would make more IgE. So, just like everywhere else, the B cell maturation requires 3 signals: recognition, activation of the BCR, interaction with the T cell receptor, co-activation through CD40, and cytokine secretion. So just like anywhere else, you get a germinal center reaction and production of effector and memory B cells, and some of those B cells are the plasma cells, which produce IgA in the gut. And I also discussed previously when we talked about IgA, the isoform IgA, that it has a specialized J chain which binds it into a dimer, and there's the poly-Ig receptor on epithelium in the gut and lung is a specific transporter of IgA from the lamina propria into the lumen. And this can also transport the IgM as well, because it also has a J chain. Okay, so more specifics than you need on IgA. Clara's pretty gung-ho about the mucosa. Okay. Interestingly, about half of IgA are cross-reactive, so they're not exclusively specific like IgGs can be. So you would think that might be a concern if they would bind self molecules, but it's not a very effective activator of complement. So IgA is not good at much of anything other than blocking and binding. So it's okay for them to have less selectivity. They're very resistant to host and microbial proteases, which you can imagine because they're working in the gut environment where we're digesting food, and there's all those non-self pathogens living there. So they're very resistant to digestion. And not only can they bind pathogens through their antigen binding site, but they're also coated by a lot of carbohydrates, which can also bind pathogens, even if it's not a specific response. Okay. So, just like in other tissues, there are cytotoxic T cell responses in the gut. In the Peyer's patches, they're found in the follicular areas surrounding the intestinal follicles. We've been through this actually in terms of the M cells capturing pathogen antigens, and the T cell responses, the cytotoxic T cell responses are like cytotoxic T cell responses elsewhere, where the effector CD8 positive cells can then recirculate and live in the lamina propria. So remember, cytotoxic T cells do kill virally infected cells or foreign cells through exocytosis. They release granules to their specific pathogen. Okay, so just like the other lymphoid tissues, lymphocytes recirculate between the mucosal associated lymphoid tissue and the bloodstream. So just like your immature lymphocytes recirculate through your lymph nodes and your spleen, leaving the circulation to the high endothelial venules, entering the lymphatics, and then emptying back through the thoracic duct. So recirculation rate is 1 to 2% per hour, so that means that in the malt, 1 to 2% of the lymphocytes are turning over every hour, so it's pretty rapid and increases the chances that specific B and T cells will encounter their antigen if it's being presented there at any given time. Just a bit more physiology here. I don't think you need to know. This is what the virus patches look like. How's that? Okay. And you don't need to know about autophagy either. Okay, so I don't think that's how you spell awry. But maybe I'm wrong. Okay, so as we've been stressing throughout this lecture, there's all that potentially pro-inflammatory tissue in the gut, and small changes in the balance of gut health, pro and anti-inflammatory effects can lead to intestinal inflammation, inflammatory bowel disease. So mutations in the NLR, NOD2, which is, again, a non-TLR pathogen recognition receptor, leads to increased activation of inflammatory responses in the gut, and increases the chance of inflammatory bowel disease. In addition, genes associated with apoptosis or autophagy, in other words, the death of cells, also increase the chance. So basically, if your gut epithelium dies a little more easily, you can have pro-inflammatory bowel disease as well. So it's just pointing out that mutations in many different molecules can alter the fine balance of inflammation in the gut, whether it's, go ahead.
Crohn's disease, does that have something to do with mutations in the lining of your intestine or your gut?
It can.
Or how, so is it directly affiliated with that, or are there other things that come into play? Because I know that Crohn's disease basically is like, it's not directly the irritation of like your gut, but isn't it correlated to that?
So just like most of the diseases we've talked about, there are genetic mutations that predispose to Crohn's but don't guarantee it. So certain mutations, like in NOD2 or autophagy genes or cytokines, increases the chance of developing Crohn's disease or ulcerative colitis. And there are hundreds of genes either associated with the epithelium or the immune system that can predispose to Crohn's or ulcerative colitis, which are basically two different variants of gut destruction.
I've also heard like Crohn's being associated or being described as having a hole in your intestines. Is that exactly what it is, or is it literally just the thinning of the lining of it, or something of that sort?
No, people with Crohn's disease have gut perforations. They get gut perforations, so it can actually penetrate all the way through not just the epithelium, but the subepithelium and the muscular layer, so that the contents of the gut can leak. And usually, a Crohn's disease patient presents for the first time because of a rupture like that, or they get adhesions between different loops of the intestine that actually blocks the passage of digestive material. So you guys are all, maybe that somebody in here has Crohn's disease, you're in sort of one of the peak ages of presentation of Crohn's disease for the first time. So this just points out what we've been talking about. How do you end up with Crohn's disease? You have a genetic predisposition, then something in the environment happens. You could have a viral infection. You could have some change in your cytokine response. You could have some change in your gut bacteria that changes the pattern recognition receptors. You could have unusual lymphocyte regulation. And you could have some event that damages your gut epithelium physically that sets off enough inflammation to trigger it. These are all different theories of Crohn's disease that have some evidence, but none of them, and probably more than one, is true.
How are these bacteria affected if, let's say, a person has a parasite like tapeworms or something in their body? Do they react differently to a person with tapeworm versus someone who does not have it?
Does the immune system respond differently?
Yeah.
Right, so tapeworms are extracellular parasites, and so this is what IgE is for. So you tend to have a lot of Th2 reactions to parasites, make a lot of IL-4 and IL-13, make IgE, and so mast cells and eosinophils then are responsible for having the immune response to clear those foreign infestations. So not too common in the Western world, but more common in other parts of the world. But it's not incredibly efficacious. We can clear lots of extracellular parasites, but we're not good at clearing tapeworms. Basically, you can harbor a tapeworm for years. Did that answer your question? Yeah.
You may have just said this in a different way that I didn't understand, but is Crohn's disease the body's inability to stop infection, or is it like a self-attacking?
So it's not autoimmunity. So
Okay.
the immune system is not attacking the self cells of the gut. It's called an auto-inflammatory disease. So basically, we're having exaggerated inflammatory responses to the contents of the gut that leads to tissue damage. So it's a cell-mediated, so remembers their hypersensitivity types. It's either 3 or 4. It's cell-mediated, but not autoimmune. Thank you, Nancy. Type 4 hypersensitivity. So we're causing tissue damage because of excessive cell-mediated immune response, but it's not against self-antigens. Okay. So we're doing well. Just mention the locations and some specifics about the other mucosal-associated lymphoid tissue, which includes nasal and bronchial-associated lymphoid tissue. This just shows the location of the adenoids and the tonsil. These are two of the important inductive immune sites. So these are basically subepithelial lymphoid follicles in your nasopharynx, ideally located to be exposed to antigens you are swallowing or inhaling. So just like I just said, they're nasal submucosal glands in the tonsils and epithelial layers of the nasopharynx. In the vault is similar structures further down the airway, the large airways. So when you inhale, the first barrier are hairs, obviously. Then, as I mentioned in one of the early lectures, there's constant mucus secretion into the bronchial tissue and the continuous movement of that mucus up towards the mouth with cilia. Larger accumulations of mucus can be cleared by coughing. So obviously, just like in the gut, the job of that mucus is to trap the invaders and prevent them from getting attachment to self, and it's also a high concentration of all those antimicrobial molecules that we talked about in the gut. So that's what somebody with tonsillitis looks like. So basically they have an inflammatory response, usually to a bacterial infection in the nasopharynx. And historically, when there was more surgery than antibiotics, people who chronically had throat infections or ear infections had their tonsils removed. That's less common these days. Okay, so the Balt inductive sites are somewhat less structured than the Galt inductive sites, but they do have specialized cells akin to the M cells, which aren't covered with cilia and are good at transporting antigens. So just like in the gut, those specialized cells transport antigens across for presentation to the immune system. So here are those cells between the ciliated epithelium, and they transport antigen into the dendritic cells underneath the epithelium for antigen presentation. And just like everywhere else, if there is enough inflammatory signal to a foreign pathogen, you get a germinal center reaction, expansion of specific B and T cells, memory and effector cells. And that's what it looks like. Okay. When things go array, you have repeated inflammatory responses, like in asthma within the lung, which changes. Those repeated inflammatory responses lead to thickening, fibrosis, and increase of the muscular layer, so blockade of the airway, which reduces your ability to inhale, and acute responses to inhaled antigen cross-links IgE on mast cells, releases histamine and other vasoconstrictors, and you get an asthma attack. If we give Nabil some dog hair, maybe we'll see one soon.
Maybe. Maybe.
Okay. We've covered that. Okay. So the last, there's the one malt site that is very unique is the vagina. So it lacks organized structures like the Balt, Nalt, and Galt. There is no lymphoid structures. There are cells of the innate immune system. There are no mucosal or systemic immune responses that happen there. So that makes sense because you don't want to kill off sperm, or some people don't want to kill off sperm. Depends on where you are in your life. But so the vagina is uniquely designed to not have immune responses to foreign substances. And so that leads to the fact that it's a somewhat immunocompromised place. So it's a good place for STDs and HIV to get in. It's a place that can be, some people have a problem with yeast infections and other chronic pathogenic infections there because it's a pretty immunocompromised place because of its function and reproduction. So penile urethra is opposite. It has the normal sorts of functions of the other malt because it's programmed to deliver, not receive. Okay. So very much like the malt is the salt, which except for the fact that it's not wet and we don't have a mucus layer, we are threatened by pathogens through the skin. Instead of having mucus and villi, we have the stratified epithelium up here, which is comprised of cells, dead cells, or cells in various stages of dying. They're very tightly linked to each other. So the live portion of the skin is the dermis, over which the epidermis is separated by the basement membrane. So there's a lot of tight junctions to prevent infection. So here's a comparison between the skin and mucosa. You can read that, but obviously there's some differences in that it's not constantly subjected to a mucus, to a pathogen load like the gut is. It's also a less appealing environment in terms of pathogens living because it's dry and acidic, but it has some commonalities. So the most important immune function of the skin is obviously as a barrier. Any of us, basically if you scratch your skin, if you break the epidermal layer and you don't clean it, in all likelihood you will get a skin infection. These happen to all of us all the time. If you get a scratch and don't clean it out, you do get a little bit of local inflammation, and that's because you do get inflamed. So obviously the physical barrier is extremely important, and that physical barrier is provided by keratinocytes. And the keratinocytes make other things besides dead cells that cover the outside. They also make digestive enzymes and fatty acids that keep the pH very low, so it's a less appealing place for bacteria to live. Nonetheless, we are constantly coated by bacteria at all times. And this picture shows all the different varieties of bugs that can be found on the skin surface, in the digestive tract, or in the oral area at any given time. Okay, so under the epidermis, getting into the dermis, the living tissue is full of inflammatory cells quite akin to the lamina propria in the gut. So we have T cells and immature dendritic cells. In the skin, they're specialized dendritic cells called Langerhans cells. Sir, sorry, somebody say something? So they do the job, same job that dendritic cells do elsewhere, to sample antigens and present. They don't have the tolerogenic function that gut epithelial cells do. So the epidermis starts with the keratinocytes at the bottom, and then there's the basement membrane, which is a very, very tight junction. And so there are no blood vessels that get through. So there's not a lot of good nutrient supply to the keratinocytes above the basement membrane. So basically penetrating below the basement membrane is what allows pathogens to enter the tissue. This is more detail than we need. So as I've implied, the pathogens have to get through the epithelium into the dermis to have an immune response. The dermis is full of those Langerhans cells and macrophages that release inflammatory cytokines. Keratinocytes can also respond. They express some TLRs. And then we have the normal, innate, and adaptive immune responses we've been describing throughout the semester. And T cells and B cells do the same things they do everywhere else. So the last piece is that IgA expressing B cells recirculate through different mucosal tissues. So if you have an immune response that results in IgA production in the gut, then the plasma cells and memory cells will recirculate to be associated with the respiratory tract and the salivary glands, so that you're making those soluble IgA into all of your mucosal tissue. So if you get a pathogen at one place, you are protected at other places with the same antibody specificity as well. and that recirculation of IgA secreting cells is dependent on a specific receptor interaction, as you might expect. I need 10 minutes. Is that okay with you guys? Okay, good. Take 10. We'll start at 20 after.
She is very needy.
Huh?
When I stop, she like put a hand on me like this.
It's too adorable.